early, so we have a 3-4 minute gap. What should we do? Hello, we're back. Next, we have Meenakshi Asokan from Princeton, and she's going to tell us about hormone-mediated reorganization of cortical dynamics during social choice.
Thank you. I'm very grateful for the opportunity to present you all my postdoctoral work in Annegret Falkner's lab. So today, I'll take you on a journey through the ways sex hormones shape social choice and underlying neural dynamics. Sex steroid hormones, such as estrogen, rise and fall over the course of the human menstrual cycle and the shorter 4 to 5 day mouse estrus cycle. Estradiol, which is the active form of estrogen, peaks right before ovulation and helps sustain costly behaviors during these optimal time windows. For example, female mice are more likely to display rejective behaviors when their hormone levels are low, and are more likely to be sexually receptive only in this periovulatory phase when the hormone levels peak. These hormones primarily act via nuclear receptors that act as transcription factors that then regulate the expression of genes, a lot of which are related to wiring and synaptic coupling. These processes are also active across sexes. So these hormones are causing major structural changes, periodically rewiring and remodeling the brain, and yet we know so little about how these wiring changes then translate to changes in neural dynamics and behavior. Also, a lot of what we know about these hormone-mediated changes in the brain are restricted to hypothalamic sites that drive these instinctual behaviors. But mating is more than just passive receptivity. The process inherently involves active choice selection and decision-making. So hormones likely modulate proactive social choice and decision-making, and these are processes that involve crucial top-down hubs such as the MPFC, the medial prefrontal cortex, which shows a modest expression of the nuclear estrogen receptors and has a well-established role in social decision-making and reward processing. But does estrogen modulate cortical dynamics and coding to regulate social choice? So the short answer is yes. And here I'll take you through a few different levels to show how hormones modulate social choices in mice and how we can track the changes in neural dynamics and coding across these long-term hormonal shifts, and how we are trying to make sense of these changes based on the underlying changes in network connectivity. We use ovary-ectomized mice with low baseline hormone levels and either inject oil or estradiol on a daily basis to reliably induce these low and high estrogenic states. And we can repeat this over multiple cycles. We took a longitudinal dense sampling approach wherein we record behavior and neural activity every day to track the changes over the course of these changing hormone levels. And we use a simple social choice assay where the female subject can choose to interact and spend time either with a male or a female conspecific tethered to the two corners in the arena. We intentionally retain the same conspecifics and the spatial location across days within a cycle to lock in the social spatial coding and really be able to attribute neural changes to the changing hormone levels. And with an 8-camera setup, you can see how we can track the poses of all 3 mice in 3D using sleep anapose. There's a lot of rich quantitative analysis in the works, and ask me about it later. But today, I'll focus on simple intuitive metrics we use to evaluate the subject's social choice preference and persistence. Here is the subject's trajectory over the entire session, and we can also look at the distance between the subject and the male and the female conspecifics and how that unfolds over time, and that's shown in the blue and the pink lines here. And this allows us to parse male and female interactions as well as self-directed behaviors as different states. We can look at the choice preference based on the percentage time spent in each state, and we can also look at the momentum and transitions between the different behavioral states, and we can quantify a choice persistence score that reflects the probability that the subject stays in a certain state once it gets there. We see that female mice are more female preferring when their estradiol levels are low, and not so surprisingly become more male preferring after the estrogen surge. Interestingly, when we look at the transition probabilities, we see that there is an increase in persistence, particularly during male interactions on the high estradiol days. So to summarize, we see that estrogen increases both male choice preference and persistence. And to our surprise, when we knock down the nuclear estrogen receptors, both ESR1 and 2 in MPFC, using a short hairpin RNA, we see that it's sufficient to abolish these changes both in preference and persistence. So the local estrogenic action in MPFC does play an important role for driving these changes in social choice. So now let's look at our NeuroPixels recordings from MPFC during these social interactions. After spike sorting, we obtain hundreds of single units from each session whose activity we can track during these social interactions and across days and across hormonal shifts. We first ask if we can discover meaningful neural states from the entire population activity. And does that correspond to the behavioral states that we have defined? So we use a hidden Markov model for detecting the neural patterns agnostic to behavior. Each hidden state is defined by a population vector of firing rates for all neurons, and in the corresponding observations, the neurons discharge as a stationary Poisson process conditional on those firing rates. And based on the Bayesian information criteria, we got the best model fit for about 4 hidden states across most days. And this model is completely naive to the behavior, but we do see a good correspondence between the neural states and the behavioral states. So we establish a dictionary between the hidden neural states and the behavioral states by tagging each neural state with the behavior that most strongly predicts its occurrence. During low estrogenic days, even on shorter time scales, we see that there are neural state switches. And we see this within all 3 behavioral states. And with estradiol, the neural state switches go down, and we see an overall increase in the neural state persistence. This could suggest that the underlying neural representations of the different behavioral states are more separable in the high estrogenic state. So to directly test that, we project the population activity in each time bin onto a lower dimensional space, here using linear discriminant analysis to maximize state separability. We then compute pairwise distances between the latent space representations of each behavioral state, and we see that estrogen enhances the population discriminability of these state representations. So far, I've shown you how estrogen enhances persistence in both behavioral choices and neural states. Next, we'll look at the underlying changes in neural coding and connectivity that may drive these shifts in population dynamics. Here, you're looking at the firing rate of a single unit across the entire behavioral session. And by zooming in around these state onsets, we can construct these rasters, and based on the unit's normalized response to the 3 behavioral states, we can then assign it a tuning and a sparseness metric that indicates how broadly or selectively tuned the unit is. When we examine sparseness across the entire population recorded on the low and the high estrogenic days, we see a shift. Units move from being more broadly tuned, clustered here near the center, to being more selectively tuned, closer to the vertices, and also interestingly towards all 3 vertices, indicating more selective tuning to all 3 behavioral states. So we see an overall increase in the percentage of neurons that are now more sparsely tuned. We then use unit match to track single units over days. And this shows the potential matches between units across 6 days of recording, where we're able to track about 50 to 100 units across consecutive days. And here is one example unit and its waveforms. And we have tracked this unit across 6 days and across these long-term hormonal shifts. We then quantified drift as a change in the tuning of the tracked units across consecutive days. And in low estrogenic state, single units that we have tracked show considerable drift in their representations of the behavioral states. And estrogen reduces this drift and enables more stable representations across consecutive days. So at the level of neural coding, we see that estrogen enhances the selectivity of tuning and reduces the drift in tuning across days. This increased selectivity may help explain why the population representations become more separable and exhibit fewer state switches. And lastly, we address how changes in network connectivity might underlie this more selective and stable tuning in the high estrogenic state. So to assess functional connectivity, we compute spike cross covariance during spontaneous spiking, and we consider a pair functionally coupled only if it exhibits a significant cross-covariance peak above the confidence interval within a 5 millisecond temporal lag. We then apply graph-theoretic metrics to assess functional coupling within the network. In this example, each node represents a single unit, each edge connects a significantly functionally coupled unit, pair of units, and the node color indicates its degree, which is a normalized measure of how connected the unit is to all the other units within the recorded population. We observe that estradiol significantly enhances network connectivity, both at the level of average node degree and also the overall network density increases. So finally, we are examining connectivity within and across ensembles tuned to specific behavioral states. And we're testing whether estradiol increases coupling more strongly within similarly tuned ensembles, potentially explaining the increase in selectively tuned units and the greater stability of representations. So if there's one takeaway, it's that hormones play a huge role in controlling this knob on plasticity and have profound effects moving networks between very different states. To summarize, we have dissected the hormone-mediated changes at multiple levels in MPFC, where we see that local estrogenic action is key to driving persistent social choice. And in line with the transcriptional changes that regulate wiring and synaptic coupling, we see that estrogen enhances functional coupling, leading to more selective and stable tuning, and that then supports persistent neural states and behavioral states. With that, I would like to extend a big thank you to everyone who made this work possible, especially Annegret for being an amazing, kind, and supportive mentor, and other people involved in the project, Stefan, Lucy, Alyssa, Dexter, and the rest of my lab for being the most supportive crew I could have asked for. Thank you all for your attention. I'm happy to take questions.
I'll ask the first question. So early on, you presented the causal relationship, right, in terms of the role of MPFC and the behavior that you're showing. My question is, from the low to high estrogen, you see the increase in persistence in behavior. Do you think the increase in stability and discriminability in the neural population is reflecting the increased stability or in behavior? What's the causal graph there?
Yeah, that's a great question. We see an increase in persistence, in the behavioral persistence, only in the male state. But when it comes to neural state switches, there's an overall increase in neural state persistence across all three states. So there are definitely probably other regions and other factors also contributing to the changes in the behavior. But we do think if we knock down the estrogen receptors and see that these changes do go away, we would also like to record neural activity with the estrogen receptors knocked down and see how the changes in neural activity then probably go in parallel with the changes in the behavior.
Oh, yeah, that would be really cool. There's some questions there. Is that not working or it needs to be turned on maybe?
Sure.
Oh, there we go.
Thanks for the great talk. I was curious, is there any coupling between estrogen and the other neuromodulatory systems, especially dopamine?
There's work from other groups, from Christine Constantinople's group, showing that there's definitely estrogen-dependent changes in activity in especially the dopamine reuptake process. And so there's definitely a crosstalk between the different systems for sure.
Hey, thanks for your nice talk. I was wondering how specific is the effect that you show to the behavior that you're looking at, to social behavior per se. I think it relates to the first question that was asked.
So the hormone-mediated changes are probably affecting more than just social behavior, or just hormones itself are affecting definitely more than just social behavior. And in this context, especially the fact that we're looking at MPFC in the context of this moment-to-moment changes in social choice, we think it's definitely directly related to the behavior. But yes, the next step would be to test if the local estrogenic action is really also causal to all the network connectivity changes and the changes in neural coding itself.
Thanks so much.
Question here?
Hey, is there an effect of this estrous state of the tethered female on any of the behaviors?
Good question. Not that we see. We track the estrous state and we don't see any changes in the behavior just depending on the tethered female's estrous state. Thanks for the question.
Since your functional connectivity is related to this very tight timing in time, I was wondering, can you speculate on why the estrogen receptor is able to affect it at that very tight time scale, basically? It doesn't feel like it's a network effect.
Yeah, interesting question. Yeah. So we do look at, we're especially looking at a 5 millisecond temporal lag when we're trying to assess functional coupling, and I don't mean to claim that it's monosynaptic necessarily. It can be recurrent changes, but yes, but the time scale of estrogen-mediated changes in general is very different. So we're not looking at membrane-bound estrogen receptors. Mostly in MPFCs, these are nuclear estrogen receptors, and it's a very different time scale. But we just think that the network is now in a different state. Either maybe there's just more recurrence, especially within the similarly tuned ensembles, that causes this increased the spike cross covariance and the increased connectivity. But yes, I don't think it's like a direct short time scale effect.
Oh, thank you.
So I'm wondering how much all of these behaviors is related to pheromones.
I'm sure there's contribution of also like pheromonal signaling, and that is also as modulated by the hormones. And probably PFC receives inputs from centers that are processing the olfactory inputs. But I think what we're seeing is more than just like any sensory representation, right? We are seeing like, especially with like neural state switches in a region like MPFC during like persistent behavior. I don't think just pheromonal signaling could explain any of that. So we are thinking of this to be just more of an internal state representation of the hormonal state itself.
Yeah, you can try and ablate maybe the auxiliary olfactory bulb and see what happens, for example.
Lisa Stowers has shown that there are changes even at a sensory level, especially with the pheromonal signaling that is mediated by changes in estrogen levels. So there's definitely going to be contribution from all these other regions, like also all the hypothalamic sites that show estrogenic modulation. So there's no doubt that those will all probably also feed into the changes that we see in MPFC.
Thank you.
Okay, thank you so much.
Sorry.
All right. Next we have Amy LeMessurier from NYU School of Medicine. Projection-specific cortical processing of vocalizations driving mouse maternal behavior.
Great, thanks for the introduction, and thanks so much to the whole program committee for putting together such a great conference and the invitation to give a talk. So, social animals of many species rely on acoustic cues to communicate across contexts, and often these vocalizations can have really similar acoustic features, but have different meanings. So it's crucial for individuals to be able to detect and categorize these sounds and to respond with the appropriate behavior in response. So for example, distinguishing calls that indicate aggression from courtship, or for caregivers, recognizing the calls of an infant in distress and caring for them as needed. So this latter case is what my research focuses on. So in altricial animals, parents' ability to respond to their infant sensory cues is crucial for their survival. So, for humans, anyone who's been around a crying baby immediately knows that this sound immediately draws their attention. So mouse pups, much like human babies, also have specific vocalizations for different needs. And one category that we study are isolation calls, which young pups emit when they are outside of the nest and their body temperature rapidly drops. And these are multisyllabic ultrasonic whistles. Here's an example right here that have a very stereotyped temporal structure. And we think that this is particularly important for responding to them. And when an experienced caretaker hears this, she will pretty much immediately leave the nest, search for the pup, find it very quickly, and bring it back. And this is done very robustly by experienced caretakers, but it is also depends on a fair amount of learning, as we can observe in virgins as they gain expertise. So we can co-house virgin females with a mom and her litter, and over several days, she'll start to retrieve pups. So we think this is a particularly interesting behavior for looking at how sensory inputs get linked with behavioral outputs, and how this is learned. So from previous work in our lab and others, we know that pup retrieval requires activity in the left auditory cortex in moms. So if you put mucimol in left auditory cortex, their ability to retrieve pups goes down pretty drastically. And this is correlated with activity in the left auditory cortex that changes in response to pop calls. It changes with experience. And you can see this in this example from Robert Liu. These are units in a dam that respond much more robustly to pop calls compared to virgins. So given that auditory cortex is required for this behavior, when is it using information about USVs? So pop retrieval is a multi-step sequence. So really the caretaker has to do a bunch of things. So first, recognize the USV. She has to be motivated to leave the nest. She has to then localize the sound, approach the pup, and finally retrieve it to the nest. And auditory information could be useful at many points during the sequence, which takes place over seconds to minutes. We also think that auditory circuits are integrating over multiple call syllables and able to identify them. So the temporal structure of USVs over hundreds of milliseconds can be used to discriminate categories of calls. The intersyllable interval, for example, alone can be used to discriminate between male, female, and pop calls. So given these distinct timescales, what are the auditory circuits that are doing these computations? So since we know that auditory cortex is needed for successful retrieval, and because of its wiring within the auditory circuit, I hypothesize that it's probably doing something important related to processing information about calls over one or both of these timescales. So auditory cortex projects very broadly to multiple areas and often reciprocally. So there are many candidates for downstream circuits linking these sounds to behavior. And I'm thinking about them in terms of top-down circuits in which cortex projects to subcortical auditory areas earlier in the feed-forward auditory pathway, and feed-forward circuits in which auditory cortex sends information about acoustics to higher-order cortical areas and the striatum. So just to further simplify this, I decided to focus on two specific projections that we know from previous work are involved in auditory perceptual learning. So projections to the inferior colliculus, which are very dense, and this is the first major multimodal processing hub for auditory input, as well as the tail of the striatum, which receives auditory input directly from primary auditory cortex and thalamus, and thus could provide a more direct link with motor sequencing circuits that could be involved in pop retrieval. And both of these projections are primarily from excitatory neurons in layer 5. So what is the circuit through which auditory cortex is facilitating this maternal behavior? So I first asked whether these projections are actually involved in pop retrieval. So to do this, we used a retrograde virus approach to target inhibitory dreads to either corticocollicular or corticostriatal projection neurons in auditory cortex. And then we tested retrieval after IP injection of either CNO or saline. So I first did this experiment using an excitatory layer 5 Cree line to just generally block most descending projections from cortex. And what we found is that retrieval was greatly impaired. So I'm showing this here in terms of percentage of pups retrieved. Surprisingly, when we selectively blocked the corticostriatal projections, we didn't see a consistent effect, no significant effect at the group level. But then, when we looked at corticocollicular projections, we again saw a big drop in performance, mirroring what we saw with layer 5 generally, and no effect in sham. So we think that the corticocollicular projections are particularly important for this behavior. And I should say this is in expert retrievers, so virgins that have been co-housed as well as dams. So now that we know that descending projections are involved in retrieval, I turned to 2-photon calcium imaging to ask how these neurons are modulated by pop calls. So I again used retrograde viruses, this time to express GCAMP separately in either the corticostriatal or corticocollicular projection, and imaged in auditory cortex layer 5 in awake virgins while playing several example calls, as well as pure tones as a control. And when I refer to calls, I'm referring to not the individual vocalizations, but actually the multisyllabic vocalization over a duration of at least a second. And I imaged the same cells longitudinally over several days, during which virgin females were first exposed to pups by co-housing with a dam in her litter and became expert retrievers. So in general, I found that neurons in both projections respond to calls, but with variable tuning and timing. So here I'm showing you some representative units from 4 example neurons and this is to 6 different calls. So some neurons look like ROI2 here, which responds very robustly to each distinct multiple syllable call, so potentially categorizing these stimuli as calls, potentially pub calls. And however, others were selective for specific calls, like ROI1, or not responsive at all, or suppressed. And when we look at the tone tuning of these neurons, we find no relationship between spectral tuning and call responsiveness, which agrees with findings from other groups. But perhaps more strikingly, I found that in the corticocollicular projections, overall activity was increased during blocks of call playback and not just immediately following stimulus presentation. So here I'm showing an example trace from a corticocollicular neuron and a corticostriatal neuron. Green is corticocollicular and corticostriatal is purple for the rest of the talk. And this is over periods of about 30 seconds, and hopefully you can appreciate that there's this very large increase in activity, not just time locked to the calls in the corticocollicular neurons during USV playback, but not during tones. And we also don't see this in the corticostriatal neurons, though there are time-locked responses to both types of stimulus. This is quantified for all cells from these animals in each group. So the green is corticocollicular, corticostriatal is purple, and then there's this heavy tail of highly active neurons during call playback in the corticocollicular. So when we looked at the responses of these neurons over days of experience, we saw that individual cells varied a lot in their response profiles to calls, potentially akin to representational drift. But when we looked at the temporal profile of these responses, we saw that at the population level, delayed responses, so responses occurring at 2 to 5 seconds following the onset of the call, were larger in corticocollicular neurons when mice became experts compared to naive. And in the middle here is an example behavior trajectory from one animal, just showing how I'm defining expert versus naive here. Okay, so we've seen that activity in cortical projection neurons seems to be modulated over multiple timescales by USVs, and that this is potentially plastic. So I wanted to understand, how does this temporal structure evolve over the central auditory pathway, and how is auditory cortex modulating activity in earlier auditory areas? So to do this with more temporal precision, I turned to neuropixels probes. So I did simultaneous recordings in auditory cortex, auditory thalamus, and inferior colliculus. And this is in awake head-fixed virgins that are hearing example calls, individual syllables played alone, as well as tones and frequency modulated sweeps with similar temporal structure to the calls. And so one thing that's popping out is that a subset of units in all of these areas are selective for syllable sequences. So I'm showing 3 example neurons here, one from auditory cortex, one from MGB, and one from IC. Oh, uh-oh. There we go. I think I clicked somewhere weird. And so each of these neurons is very robustly responding to the second syllable of this call, but not the first. And if we can zoom in on that, and if we then play the second syllable in isolation, the responses largely go away, suggesting that these neurons are only responding to this syllable in the context of a larger sequence. And so this is potentially a mechanism for which calls could be categorized based on temporal features. Of course, this is not how all cells respond. There's diversity of responses, and so if we cluster these responses using PCA and k-means, we can see that these pretty distinct motifs fall out of this, which I'm going to qualitatively call facilitating responses, syllable locked, suppressed, onset, and delayed, including these later syllable responders. And so these are some example units on the bottom. And so when I looked at how these units are distributed across the areas that I recorded from, I found that each of these clusters are present in all 3 areas, which suggests a more complicated picture than a simple feed-forward computation, and that these could represent distinct circuits for different computations, potentially at different timescales. So one of the questions that I posed earlier was, how does auditory cortex influence timescales of responses in earlier auditory areas? And based on what we've seen so far, I hypothesized that it could be shaping responses in IC. So to test this, I'm using optogenetics to inactivate corticocollicular neurons specifically during call presentations and ask what happens in IC. So this is still in an early phase, but we're seeing hints that if we inactivate during calls for different durations, we see changes in the temporal structure of responses in some IC neurons. So to sum up, I've shown you that cortical projections to inferior colliculus are needed for pop retrieval in response to infant cries. Auditory cortex projection neurons are modulated by multisyllabic calls over multiple timescales. And we see common response motifs across recurrently connected auditory areas, which may enable categorization based on temporal structure. So with that, I'd like to acknowledge everyone in the Froemke lab, and in particular, I've had help from some excellent technicians and undergrads, especially Julia Paraiso and Gurket Kaur, and thanks to Rob as well, of course. And all of this work has been funded entirely by the NIH and the Brain Initiative, so thanks very much to funding sources, and happy to take questions.
We have time for one quick question. Go ahead.
Maybe not so quick. Okay. So you're describing auditory cues which make up what might be called pup cues for maternal behaviors, I think. And I guess you're probably familiar with the role of MPOA in controlling maternal behaviors. Mm-hmm. I was wondering, I think what you're talking about are upstream effects to MPOA, and then I was also wondering about, MPOA exists in a kind of a opponent behavior to infanticide behaviors of these animals, and wondering whether your auditory pathways are also contributing to infanticide behaviors.
Yeah, so for, as far as the first question, yeah, I think, many pup sensory cues are upstream of, hypothalamic circuits that are controlling behavior, maternal behavior. And I'm really interested in how, in particular, this auditory information is getting there. And, there's evidence from work from a former postdoc in the Froemke lab that, projections from inferior colliculus to higher order thalamus are what sort of deliver information about calls to oxytocinergic neurons in hypothalamus. And I find that to be a really interesting circuit because, I showed this complicated diagram of many connections within the auditory circuit, but that could be represent a subset of that circuit. And in terms of infanticide, yeah, I'd be curious. I'm not sure how much, the acoustic input controls that behavior. I guess you can imagine that, it is a very aversive cue, so maybe that, in non-maternal, non-parental animals, it does trigger aversive behavior, and maybe that could lead to infanticide, but yeah, it's an interesting question.
Thank you. Last talk of the session, we have our own Yaara Lefler from Sainsbury Wellcome Center, and she'll tell us about periaqueductal gray neurons are all or none threat gates modulated by escape experience.
Thanks, Anne, and thanks for the opportunity to present my work here today. So I'm going to talk about escape. which is a universal behavior that is essential for survival and that we use as a model to study how the brain generates behavior. Now we think it's a powerful model because escape is consisted of several distinct and generic steps, for example, decision making and action selection. And despite being innate, it can be flexibly changed by experience to adapt to changes in the environment or internal state of the animal. So we study escape in the lab by presenting mice with threatening stimuli, in this case a lumen stimulus, and expanding dark disc projected from above. In response, mice immediately escape to the shelter. Previous work from the lab and from others have shown that there are two midbrain areas that are key for the computation of escape behavior: the superior colliculus, which integrate the sensory threat information, and the periaqueductal gray, or PAG, which threshold this threat information to compute escape initiation. In this study, we wanted to ask how PAG neurons integrate these synaptic inputs in order to compute escape initiation. And to do that, we had to first gain intracellular access to PAG neurons during escape behavior. So we established a head fixation system, which is consisting of an hair table and a floating lightweight circular arena with a shelter zone. And we project mice with threatening stimuli using monitor and speakers positioned above the mouse. Now, mice are head fixed, but they can move, and they move the arena underneath them so that they can explore and navigate this physical environment. And they do this in similar pattern as freely moving mice in an open field arena, as you can see here with those example trajectories and heat maps of a 1 hour session. We then present mice with threatening stimuli, in this case 3 repeats of an auditory stimulus, and mice immediately escape to the shelter and stay there. And those escape trajectories are similar to the ones of freely moving mice in an open field arena. Overall, this is a very robust paradigm to elicit escape behavior in head-fixed mice. Here are speed traces from 79 trials of multiple mice. And with this paradigm in hand, we could perform wholesale patch clamp recordings. So we targeted the PAG and recorded from over 150 neurons during escape behavior. Okay, so what do we find? The first thing we see is that the membrane potential of PAG neurons has a very narrow distribution that rarely crosses the spike threshold. So they sit under a very tight control, and this makes sense because when those cells spike, mice escape, and we wouldn't want to be escaping all the time, right? So what will make those neurons spike? To check that, we first injected current steps and looked at the generation of spikes, and we saw that those cells sit on average 15 millivolts away from their spike threshold and only need around 50 picoamperes in order to elicit a spike, which is a very small amount of current that suggested to us that those cells have a very high intrinsic excitability. So we next measured the spontaneous excitatory synaptic input to those cells, here marked in arrows. And we saw that the average unitary size of synaptic potentials was of about 1 millivolt and of synaptic currents of approximately 20 picoamperes. And again, suggesting to us that those cells, because that they have high intrinsic excitability, and because their inputs are relatively large, that only a small amount of input would make those neurons reach their action potential threshold. All right, so what happens during threat? When we present threat, we see that the membrane voltage rises and stays on in a step-like response. And this is consistent across many cells. Here are voltage traces of many trials, and the average voltage, which shows a distinct depolarization step that brings the membrane potential to a higher voltage state. We call this an on response. Now, this state switch coincides with escape onset. So the peak of the average voltage aligns precisely with escape initiation, which sorry, which then, and what then happens is that those cells, because they are very close to their action potential threshold, and now the probability of spiking is higher, and thus sufficient to drive an escape response. All right, and why does this happen? Because we see that the synaptic rate increases and stays high. Overall, we saw a 20 hertz increase in synaptic rate during threat presentation. And again, this is a very small input, but because of the high excitability of PEG neurons, it is enough to reach action potential threshold and initiate an escape response. All right, now escape is a probabilistic behavior, and especially for low levels of threat, in some trials, mice did not escape, even though there is a threatening stimulus. So we looked at those trials, and we saw that the majority of cells were silent, so their voltage during the threat did not differ much from baseline. This suggested to us that PEG cells has two modes of operation, either on off or off mode with no intermediate responses. To look at this further, we looked at the small fractions of cells that showed a significant response during no escape trials, which was very similar to the on responses during escape trials, again suggesting that PEG cells has either an on or an off mode. So then what happens during escape? We see that the fraction of on cells during escape trials was much larger than no escape trials. So overall, when escape is to be initiated, more cells are turned on, rather than just changing the magnitude of individual responses. To further confirm this, we expose mice to threat with different features, either visual or auditory or both. And we first looked at superior colliculus cells, the main threat input to the PEG, and we saw that their membrane voltage dynamics reflected the different threat features. In contrast, PEG neurons responded invariantly to the different threat types and showed a uniform on response, suggesting that PEG mostly encode actionable threat, so threat that needs to be escaped from. All right, so I mentioned in the beginning that escape behavior can be modulated by experience. So how can this be implemented in our system? To study this, we developed a threat sensitization paradigm in which we present mice with repeated high-intensity threat stimuli, and a sensitization is formed with only one threat presentation. So here are speed traces of 20 mice for their first presentation of threat, and this is the average speed. We then present threat again for the same mice after this sensitization, and we see a higher vigor of response and a 40% increase in escape probability. How does this implemented in PEG neurons? 40% of the cells we recorded from had a sustained depolarization that lasted for many minutes after the offset of the threat stimulus. And in this example, it brings the cell 7 millivolts higher than baseline, locking it to a higher value that is much closer to the action potential threshold. And overall, we saw a sustained depolarization of an average of 4 millivolts. This sustained depolarization increased the excitability of PEG neurons even further. So when we inject current steps and look at the voltage generation, we see before sensitization and after the sensitization, we see that more spikes are generated for the same current pulse. And this has been seen in all the cells we recorded from. And finally, the sensitization also generates more spikes on further threat stimuli. So this is an example. And overall, in almost all the cells we recorded from, we saw that more spikes are generated to further threat stimuli after the sensitization. All right, so to summarize, I showed you that PEG neurons are quiet during exploration, and that when a threat comes, they can convert a very small amount of input into an all or none step response. And the fraction of on cells will determine whether an escape will be initiated or not. Now, with the experience of threat, PAG cells become more depolarized, such that subsequent threat, for example, in a high predatory risk environment, will generate more spikes on the on response and overall increases the vigor and probability of escape. Thank you for your attention and for all the people and funders supported this project.
We have time for a few questions.
Hi, quick question about the sustained depolarization. How much variability do you see in the amount of sustained depolarization? And does that in any way correspond to the amount of threat sensitization you see?
Yeah, so there is not much variability. I would say all of them sits around 4 millivolt plus minus.
Sorry, I meant the amount of time. You said it lasts for several minutes, so I wonder.
Yeah, so time, so when we record, we can't always keep the cells for that long, right? So we see in many cases that it's long, but we cannot measure how long it is. We also usually give another threat stimuli in order to see how it's affecting, how the sustained depolarization affects it. But it seems pretty stable over time. Regarding the second question, it's a good question, but since we record from single neurons, we can't really say anything. I mean, I believe many of the cells do that, and then exactly to correlate between single cell to the behavior, it would be, I guess, impossible. Yeah, thank you.
Question here?
Great, thanks. That was a really great talk. So what do you think these individual PAG neurons, what are they actually signaling? Is it some sub-feature of that looming stimulus that it's picking up on, and then together they collude to say, okay, this is an actual threat?
So yeah, I think, yeah, they just, whenever there is a threat, they say, oh, it's a threat. I don't care if it's a looming stimulus or an auditory stimulus. I just think it's something threatening that I need to escape from, right?
Right, so, but why would there be like so many of them? Are they trying to kind of pick up different, are they responding to different features in the sensory stimuli, or?
I don't think so. I think I don't know, maybe if some of the stimuli are more threatening than others, then you will have more cells that are wired, because we see that the input is the main thing that changes whether a cell would be an on cell or not, right? So maybe they are wired in some way that more threatening stimuli will evoke an on response in more PAG neurons, but which I don't know if I can check it myself with this method. But overall, they would represent the threat itself, right? I'm not sure if I answered your question.
That's like we can chat offline.
Okay,
Thank
perfect.
you.
One last quick question.
I'm curious about the mechanism for the all or none nature of these responses. Is anything known about the lateral interactions between these neurons?
No, we know there is a recurrency in the superior colliculus. I'm not sure we know anything about the PAG. Are you asking specifically about the sustained depolarization, or?
Also, but primarily, it seems like the whole PAG either lights up or doesn't, and so somehow you want to coordinate the activity of those neurons.
So I believe it comes from the input already that brings the threat information. I'm not sure if it's related to the way they wire up. I don't know, nothing that I can say about it. I can say that we see that the sustained depolarization is most likely arising from disinhibition of inhibitory neurons in the PAG. So this might be something that works there to collaborate between them. Yeah.
All right,
Thanks.
Let's thank all the speakers from this session. We have a coffee break now, and the next session starts at 6:15.